The kidneys filter blood plasma, removing metabolic wastes toxins from the body, and excrete them in the form of urine. During this process, they also maintain constant volume and composition of the blood, or homeostasis. Blood enters the kidney via the renal artery, which divides to smaller arteries and finally arterioles. The arterioles get into contact with functional units of the kidney called nephrons. This is where blood filtration and urine formation take place. The filtered blood is then collected into a series of larger veins and exits the kidney through the renal vein. The urine is collected in collecting ducts and leaves the kidney via the ureters. Each kidney contains over 1 million nephrons. A nephron is composed of two major parts, a capsule known as glomerular capsule, or Bowman's capsule, and a long renal tubule. Renal tubules of several nephrons connect to a common collecting duct. There are 3 steps in the formation of urine. Glomerular filtration takes place in the Bowman's capsule. Tubular reabsorption and secretion occur in the renal tubule. Water conservation happens in the collecting duct. Blood enters the Bowman's capsule via the afferent arteriole, passes through a ball of capillaries called the glomerulus, and leaves via the efferent arteriole. The afferent arteriole is significantly larger than the efferent arteriole, creating a blood flow with a large inlet and small outlet. As a result, the blood hydrostatic pressure in these capillaries is much higher than normal. Hydrostatic and osmotic pressures drive water and solutes from blood plasma through a filtration membrane into the capsular space of nephron. The filtration membrane acts like a sieve, allowing only small molecules to pass through. These include water, inorganic ions, glucose, amino acids, and various metabolic wastes such as urea and creatinine. This fluid is called glomerular filtrate. The amount of filtrate produced per minute is called glomerular filtration rate, or GFR. The GFR is kept at a stable value by several feedback mechanisms within the kidneys. This is known as renal autoregulation. The GFR is also under sympathetic and hormonal control. GFR control is generally achieved by constriction or dilation of the afferent arteriole, which causes the glomerular blood pressure to fall or rise, respectively. In a healthy person, the total filtrate volume amounts between 150 and 180 liters a day. However, only about 1% of this is excreted as urine, the rest 99% is reabsorbed back to the blood as the filtrate flows through the long renal tubule. This is possible because the efferent arteriole, after exiting the Bowman's capsule, branches out to form a network of capillaries, known as peritubular capillaries, which surround the renal tubule. The first part of the renal tubule, the proximal convoluted tubule, reabsorbs about 2/3 of the filtrate. In this process, water and solutes are driven through the epithelial cells that line the tubule, into the extracellular space. They are then taken up by the peritubular capillaries. Sodium reabsorption is most important, as it creates osmotic pressure that drives water, and electrical gradient that drives negatively charged ions. Sodium level inside the epithelial cells is kept low thanks to the sodium-potassium pumps that constantly pump sodium ions out into the extracellular space. This creates a concentration gradient that favors sodium diffusion from tubular fluid into the cells. Sodium is absorbed by some port proteins that also bind glucose and some other solutes. Nearly all glucose and amino acids are reabsorbed back to the blood at this stage. About half of nitrogenous wastes also reabsorbs back to the bloodstream. The kidneys reduce the blood levels of metabolic wastes to a safe amount, but do not completely eliminate them. Some of the reabsorption also occurs by the paracellular route, through tight junctions between the epithelial cells. At the same time, tubular secretion, where additional wastes, drugs and other solutes leave the bloodstream to join the tubular fluid, also takes place. The processes of reabsorption and secretion continue in the nephron loop, the loop of Henle, and the distal convoluted tubule. However, these parts of the tubule also have some other important functions. The main function of the loop of Henle is to create and maintain an osmolarity gradient in the medulla, that enables the collecting ducts to concentrate urine at a later stage. The ascending limb of the loop actively pumps sodium out, making the medulla salty. The descending limb of the loop is permeable to water but much less to sodium. As the water exits the tubule by osmosis, the filtrate gets more and more concentrated as it reaches the bottom. The ascending limb, on the other hand, is permeable to ions but not water. As a result, the filtrate loses sodium as it goes up and becomes more diluted at the top of the loop. The medulla is in equilibrium with the loop and hence has the same salinity gradient, more salt at the bottom. Reabsorption and secretion in the distal convoluted tubule are under control of various hormones. This is how the kidneys respond to the body's needs and adjust the composition of urine accordingly. The collecting duct receives tubular fluid from several nephrons. The main function of the collecting duct is to concentrate urine, and therefore conserve water. This is made possible by the osmolarity gradient generated by the loop of Henle. As it gets saltier deep in the medulla, the filtrate loses more and more water as it flows down the collecting duct. The collecting duct is also under hormonal control, so it can adjust the amount of reabsorbed water accordingly to the body's state of hydration. For example, when the body is dehydrated, more water is reabsorbed back to the blood and a small volume of excreted urine is more concentrated.